Hey guys, in this video we're going to be talking about meiosis for the most part, but we're also going to be talking about the difference between meiosis and mitosis. All right, so to start that off, let's kind of just remember a few things. The first thing we should be talking about is the idea of different types of cells in the body. Your body is made up of cells called somatic cells, and there's a few cells in your body called gametes. So a somatic cell is things like your skin or your bones or your nerves or your muscles. Those are all somatic cells, and they all have a full set of DNA, which we call diploid, and we designate as 2 ends. So in humans, here's a karyotype of a human, we would designate that with 46 chromosomes. Now, a gamete is a reproduction cell, so it's like a sperm or an egg. Those cells do not have a full set, they only have a half set of DNA, so we would call them haploid, okay? So in terms of how we make each of these cells, mitosis is used to make somatic cells, and meiosis is used to make gametes. Let's kind of roll through the situation together, okay? They both start the same, except that meiosis starts with a special cell called a germ cell. So meiosis starts with a germ cell. Both of them replicate their DNA, okay? So we go to a double the DNA during the S phase. Both of them do that. All right, so now we are ready finally to do our first PMAT. Okay, so they both go through prophase, metaphase, anaphase, telophase, and they split their nuclei and their chromosomes. Now they do this a little differently. So take a look at this, guys. For mitosis, you can see that there's a splitting of the sister chromatids. So there they go, and they each go to different sides. What that means is we're going to get 2 cells in the end that are duplicates of each other. They are clones, and not only of each other, but they're also clones to the original cell, okay? If we think about what that looks like with our chromosome models, it's going to look something like this, all right? So here we have our chromosome models, all right? And in mitosis, the following takes place. We actually split them up. We get a mom and a dad go one way, and a mom and a dad go the other way. This is why both of them are considered somatic cells, because they're both diploid, okay? Let's finish off with mitosis. All right, so to finish off mitosis, we can see that we're making diploid somatic cells, and they're genetically identical. They are clones of each other. Now in meiosis, it's a little different. You can see that the end result is 2 cells that each are haploid, but they're like double haploid. Now what's going on here? Well, first thing we have to understand is that crossing over is taking place. Crossing over is when a tetrad is formed, and there's a little bit of mom and a little bit of dad DNA that kind of cross with each other. So let's take a look at what that looks like, guys. So during prophase 1, we can see that the homologous chromosomes get so close to each other that they form something called a tetrad. And in that tetrad, we can see that they actually get so close that they switch a little bit of DNA. So it's a little bit of the mom and a little bit of DNA switch each other, okay? Then in metaphase, watch what happens. Instead of sister chromatids separating, we see that homologous chromosomes separate. This is only a dad and this is only a mom, so it's not a full set of DNA. All right, so now meiosis is not finished. Meiosis has to go through another, so here's a graphic of what's going on there. There we see homologous chromosomes after crossing over are moving away from each other. All right, so meiosis is not done. It goes through another split. So meiosis 2 takes place, and here in meiosis 2, we can see that the sister chromatids are now going to separate. In our final result, you can see that we have 4 cells. They're genetically unique from each other. That's why all those are different colors down there. They're all considered to be haploid, okay? And these are our sex cells, right? This is how we've now made gametes. We've now made either sperm or eggs, okay? Now, problems can happen during this. For example, something called nondisjunction. If this happens to a gamete, it's going to affect the offspring. Now, this could happen during mitosis too, but if it happens during mitosis, it'll only affect the parent. If it happens to the gamete, it's going to affect the offspring. Okay? Nondisjunction is when we don't have the spindles attaching correctly, and it creates errors like trisomy and monosomy. All right, guys, if you have any confusion about mitosis or meiosis or these big idea concepts, you should re-watch this video.